Hello, everyone, and welcome to today's webinar titled Spatial Mapping of Pain-Associated G Protein Coupled Receptors and Biomarker Localization in Mouse Brain using RNAscope, HiPlex v2, and RNA-Protein Co-Detection Assay. This webinar is a part of the 11th annual event in the Neuroscience Virtual Event Series. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by ACD Bio, a Biotechne company. For more information about ACD Bio, a Biotechne company, go to acdbio.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Sayantani Basak, Application Scientist, Advanced Cell Diagnostics at Biotechne. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Dr. Basak, you may now begin your presentation.
Hello everyone, thank you for joining my talk today. Today I'll be presenting a project on spatial mapping of G protein coupled receptors in mouse brain using RNAscope HiPlex v2 and RNA-Protein co-detection assay. So here is an overview of my presentation today. First, I will give an introduction on the RNAscope technology and some of the research areas supported by RNAscope, including neuroscience research. Then I'll talk about my project on spatial mapping of G protein coupled receptors in the mouse brain. And finally, I will summarize the study and give some important highlights on the findings. As we all know, complex, highly heterogeneous tissues such as the brain and tumors, as well as some processes such as embryonic development, are comprised of multiple cell types and states. Precise transcriptomic characterization of these tissues can enable identification of new cell types, predictive biomarkers, new therapeutic strategies, and more. However, interrogation of these complex, heterogeneous tissues require a highly sensitive and specific multiplexed spatial approach with single cell resolution. RNAscope, which is a universal in situ hybridization platform, was designed to overcome limitations of traditional-ish assays while providing highly sensitive detection of RNA biomarkers. This technology can be applied to any gene, any species, and any tissue. This assay has a unique proprietary double Z probe design and amplification strategy that can detect target mRNA transcripts at single cell and single molecule resolution with spatial context within cells and tissues. There are three key elements of this assay: the double Z probe design, signal amplification strategy, and detection technology. And this is an ideal spatial biology solution to interrogate complex tissues. The RNAscope amplification technology is highly sensitive and specific and gives a very high signal to noise ratio. The signal amplification system is based on ACD's patented double Z probes. These double Z probes need to bind in tandem on the target sequence in order to build the signal amplification cascade. As seen in this schematic, the double Z probes have a target binding site spanning approximately 50 bases, a linker region, and a pre-amplifier binding site. Once the double Z are bound to the target, the pre-amplifiers bind, followed by the amplifiers, and the amplification tree is formed. The amplifier is followed by a binding of labeled probes, chromogenic or enzyme fluorophores, which bind specifically to the amplifier region. This imparts high sensitivity for detection, as each amplification tree is specific for each double Z probe. The reaction is highly specific because if the double Z probes are not bound in tandem, the reaction is the amplification tree is not generated, and because the double Z probes need to bind in tandem in order for for the reaction to occur. So this confers specificity to the assay and minimizes off-target effects due to non-specific binding. So taken together, this combination of ACD's patented probe design and signal amplification system ensures a high signal-to-noise ratio. This slide shows the snapshot of all of our assays and applications for detecting key RNA biomarkers. We have various assays which can be used to detect different types of RNA biomarkers, each detecting specific target length and sequence. So we have RNAscope, BaseScope, and microRNAscope, and specific applications for each product. For example, with the RNAscope, you can detect RNA biomarkers of more than 300 nucleotide in length, and this comprises of all the messenger RNA and the long non-coding RNA. You can visualize subcellular RNA localization and track CAR T cells in preclinical and clinical research tissues by detecting CAR UTRs. And you can also measure immune response by detecting cytokine and chemokine expression using the RNAscope technology. The BaseScope detects smaller targets of 50 to 300 nucleotide in length, and this comprises detecting the splice variants and point mutations. This is very useful for detecting CDR sequences on TCRs, visualize oncogenic splice variants, identify circular RNAs in developing brain, and identify point mutations. With the microRNAscope, which can detect even smaller targets of 17 to 50 nucleotide in length, you can detect the biodistribution of siRNA and ASO therapeutics in the cell, and also study disease and development associated with microRNA expression and the regulation of RNA targets. So together we enable a large number of RNA and now DNA targets to this technology platform. Currently, we have over 40,000 unique pre-designed probes in several key application areas like neuroscience, cancer, stem cell, infectious diseases, etc. So you can combine any desired target probes, and we are not restricted to pre-validated panels. We can also custom design any gene for any species with any sample. We have a universal pre-treatment condition for all probes, which makes it very easy for assay optimization. And you can also perform multiplexing on FFP tissues using FFP reagent, which can control for the autofluorescence. And finally, we have assays for detecting low abundance targets using the TSA chemistry based assays. RNAscope assay has been extensively used in neuroscience research owing to its very highly specific and sensitivity in detecting gene expression in central and peripheral nervous system. So there are several application areas in which RNAscope has been used in the neuroscience research. And one such area is to detect GPCR, ion channel, and non-long coding RNAs where no reliable antibodies are available. So to visualize these molecules, researchers have extensively used RNAscope in the brain and in the peripheral nervous system. The other application that is very unique is validating the RNA-seq signature in single cells. So performing the single cell RNA sequencing and downstream validation using RNAscope assay has been a major application area. And then visualizing cellular activity in cell type specific manners, for example, plasticity markers, secreted factors, and co-localization using co-detection of protein and RNA in these cells. And then characterizing specific genetic variations, for example, alternative splicing and circular RNA in developing brain and in other disease conditions. Here is a quick overview of our RNA HiPlex V2 assay. With HiPlex V2 assay, we can do spatial mapping of 12 targets in FFP tissues and up to 48 targets in fresh frozen tissues. Currently, we can do image registration and background subtraction for ROI analysis, and the ROI data can be utilized to do image analysis using standard image analysis softwares. In the following slides, I'll be introducing our upcoming HiPlex V2 whole slide image registration capabilities. I'll be presenting data from a project that we conducted at ACD, which demonstrates how spatial mapping of GPCR family receptors and downstream image analysis is performed using RNAscope HiPlex V2 assay and the new whole slide image registration software, which is coming soon. Here's a quick background on the project that we conducted. The G protein coupled receptors, as we know, are an important component of pain modulation, and they are widely distributed in the peripheral and central nervous system, and they are one of the most important therapeutic targets in pain medicine. However, the detection of GPCRs can be challenging due to difficulties in obtaining suitable antigen accessibility and antibody production and their low expression levels. So the goal of this study is to demonstrate spatial mapping of GPCRs and cell type specific association of neuropeptide Y, which is known to be involved in pain perception and transmission in the mouse brain, using the RNAscope HiPlex V2 and the integrated code detection workflow. So the aim of the project is to spatially map GPCR family receptors, including the pain receptors, across different functional regions of the normal mouse brain using the HiPlex RNAscope assay in order to understand the spatial profiling and the spatial network between these GPCRs. And we did two different studies in order to achieve this aim. The study one was to investigate differential expression of opioid receptors and neuropeptide Y using an integrated co-detection multiomic assay in specific ROIs in different functional regions of the brain. And the second study was to analyze the expression pattern of the GPCR targets using the new HiPlex whole slide image registration platform and the image analysis software. So in the first study, we investigated simultaneous RNA and protein co-detection of the opioid receptors and NPY in specific functional regions. Just to introduce and give a little bit background on NPY, NPY is one of the most abundant neuropeptides in the CNS and is known to be involved in chronic pain. It is synthesized in the GABAergic neurons, and it acts as a neurotransmitter during cellular communication. And so we performed integrated co-detection workflow, which is short formed as ICW, to visualize the cellular source of NPY and its association with opioid receptors in microglia, astrocytes, and neurons. So here is a workflow of the ICW assay. So first we permeabilize the cells or the tissues, and then we bind the IHC antibodies first, followed by hybridization of the RNA probes, and then we amplify the RNA signal. This is followed by a secondary antibody, which then binds to our target antibodies, and then we visualize and we can quantify the ROIs. So here we did three different experiments using different antibody and RNA biomarkers, and we'll be focusing on the first two, which is the GFAP antibody, and then the other one was a NUN antibody. And then we performed the multiomic ICW with OPRM1, OPRT1, and OPRK1, which is the different opioid receptors, as well as NPY and AIF1, the microglial marker, in different experiments. So here is our co-detection image of neuronal marker NUN antibody with OPRM1, OPRK1, and OPRT1 RNA biomarkers. As you can see in the top panel, you are seeing a low mag image showing the NUN antibody along with the three different opioid receptors. And in the bottom panel, you can see the zoomed in image of each of these opioid receptors, OPRM1 in green, OPRD1 in red, and OPRK1. And some of the cells you can see the overlay of all the three opioid receptors in the neurons. So this is a qualitative image, and we quantified the different phenotypes using ROI analysis of this image. And what we were looking at is we were looking at the positive phenotypes or co-detection for either of these opioid receptors with the neuron, or amongst themselves, or like the expression of all the three receptors together as phenotypes. And we concentrated on two different regions of the brain which are important in pain modulation: the nucleus accumbens region and the thalamus. And what we saw that there was not much difference in the nucleus accumbens and the thalamus in terms of the phenotype, different phenotypes of the opioid receptors along with the neuron. So we would keep this in mind later on as we move along our analysis, and we'll try to evaluate what does this mean in terms of the distribution of these opioid receptors. This slide is showing the whole slide image co-detection of NPY with opioid receptors and microglia and neurons. So here in the left panel, you can see the GFAP antibody along with OPRM1, NPY, and AIF1, the microglial marker. And in the bottom panel, you can see how the NPY is distributed over the cortex, the striatum, as well as the hippocampus region. And we also performed another experiment with NUN antibody and NPY, GFAP and AIF1. As you can see that the GFAP RNA and protein co-localizes. So, and then we also saw the similar NPY distribution when we perform NUN antibody staining along with the other RNA biomarkers. So from the previous experiment, we selected ROIs, and we were interested to look at the nucleus accumbens and the hippocampus region, known to be important in pain regulation. So what we wanted to look at was how does NPY co-localize with the neuron or the astrocytes or the opioid receptors. So we selected 6 different ROIs, and we tried to look for co-localization of NPY with these with the other cells. And what we found that the NPY does not co-localize with the astrocytes, whereas it co-localizes with AIF1. So here is the zoomed in image of how you can see the co-localization of NPY with OPRM1, but it does not co-localize with GFAP on the top panel, and then you can see the NPY co-localized with microglia. So when we did a quantification of this ROI method between hippocampus and nucleus accumbens, we looked at the positive cells and also the positive phenotypes by co-detection. So when we looked at the positive cells, what we saw that the NPY number of positive NPY positive cells were relatively lower compared to the OPRM1 and the microglia and the astrocytes in both hippocampus and nucleus accumbens. Whereas in the positive person phenotypes, you do see that on the top panel, you do see that the NPY co-localizes with AIF1 and OPRM1, but not with the GFAP. So this suggests that the NPY and the OPRM1 do co-localize together. So to summarize the study 1, we performed simultaneous RNA and protein co-detection that indicated co-localization of NPY with opioid receptor, microglia, and neuron, but not astrocytes of the nucleus accumbens and the hippocampus. This study suggests functional insight of brain regulation in specific regions, and RNA-protein co-detection allowed for ROI-based analysis. So to evaluate more than 4 targets and expand the scope of the study to map all the GPCRs with a focus on the opioid receptors, OPRM1, OPRD1, and OPRK1, we utilize the HiPlex assay with whole slide image registration for better spatial analysis. In the second study, we utilized HiPlex assay for simultaneous visualization of 12 targets in mouse brain. HiPlex is a powerful and efficient plexing tool for visualizing and validating multiple targets. And here is our assay workflow. After a series of specific and effective signal amplification steps, the signal is detected iteratively where up to 4 target genes are visualized in 4 distinct channels. And similar to our previous RNAscope assay, a single RNA transcript can be visualized as a punctate dot. The assay uniquely employs the 4 channel of different fluorophores, and the iterative process is to be repeated until all the 12 targets are detected. The fluorophore cleaving procedure is rapid and it's minimal with no effect on the RNA and tissue morphology, since it does not strip away the bound probe. Finally, the image from various rounds can be merged using the RNAscope HiPlex image registration software. The software allows to toggle and overlay expression of only the selected targets, thereby making data interpretation very simple and easy. The goal of this study was to map the different GPCRs and pain receptors across functional regions of the brain using the new whole slide image registration platform, and then perform downstream analysis to understand cell neighborhoods. So we performed a HiPlex assay using sagittal mouse brain sections, and so we took the sagittal section and then we did our HiPlex assay, whole slide image registration and background subtraction, followed by downstream image analysis. So we used the HiPlex assay and 12 target probes against neuronal marker, neurotransmitters, opioid receptors, and other GPCR family genes. And we clustered them in different GPCR families like opioid receptors, dopamine receptors, GABAergic, and so on. And we performed the analysis on the whole slide image analysis, whole slide image, and we selected 3 regions that were annotated, which is thalamus, hippocampus, and striatum, to understand the spatial distribution of these opioid receptors in these regions. Here is a HiPlex V2 whole slide image after image registration and background subtraction. As you can see that this is showing all the 12 targets that we performed in the HiPlex V2 assay, and you can clearly see the differential expression of these 12 targets in this region of the mouse brain. This is showing a very nice clear punctate staining, what you should be seeing using the HiPlex V2 assay, and you can clearly distinguish between the differential expression patterns among the various genes in this image. The advantage of this analysis is that after you perform the image registration and background subtraction, you can directly export it to the open source image viewer like QuPath, where you can start to do the image analysis right away. Or you can use other image analysis platforms like Halo from Indica Labs, which I'll be showing in a minute. And then this makes the image analysis seamlessly connected to the whole slide image registration and background subtraction. We were interested in looking at the differential expression of the opioid receptors in the different mouse brain regions. So for this, we annotated 3 regions: the nucleus accumbens region, the thalamus, and the hippocampus. So in the left panel, you can see the expression of all the 12 GPCRs in these 3 regions of the brain. And so we wanted to specifically look at the opioid receptors. And so in the right panel, you can see the distribution of the 3 opioid receptors in these regions of the brain. So on the top right panel, as you can see that there is robust expression of all 3 opioid receptors, OPRM1, OPRK1, and OPRD1 in the nucleus accumbens region. And you can see a more or less homogeneous distribution of these 3 receptors in the nucleus accumbens regions. But when you look at the bottom panel, in the hippocampus and the thalamus region, you do see a differential expression of the opioid receptors. So in the hippocampus, you do specifically see an enrichment of OPRK1 and you don't see any other opioid receptor expression. Whereas in the thalamus, you can distinctly see a region which has high expression of OPR-D1 receptors, but then there are other regions which had a distribution of OPRM1 and OPRK1. So overall, this showed us the distribution of the opioid receptors, and we wanted to quantify and understand the spatial analysis and the neighborhood enrichment analysis between these opioid receptors in these three regions. We performed the HiPlex spatial neighborhood analysis, and a spatial map was created, or a spatial graph was created with cells as nodes and neighborhood relations between spots as the edges. So we were interested in looking at which cells are connected and which cells are not connected. So as you can see in the striatum, we saw robust expression of all three opioid receptors as we had seen before. But if you look at the granular level, you could really see the clusters of cells which are connected to one another, and then we could see the spatial relationship between those cells. In the hippocampus region, as we saw previously, we saw high expression of OPRK1 receptors, and they were like almost enriched among themselves, and we didn't see much of the other connection with other cell types expressing the other opioid receptors. In the thalamus also, you could see clear clusters of OPRM1 or OPRD1 family receptors, and then the spatial graph could show us these granular details of how these are spatially connected with each other. And so coming back to our previous phenotype co-detection that we performed using the ROI analysis, we could not see any difference in the phenotypes between these two regions, the nucleus accumbens and the thalamus. But the whole slide image registration analysis could give us a more granular level of identification and understanding of the spatial signatures and how these cells are connected with each other. We also looked at the gene expression profile of these GPCR receptors in the different regions. So irrespective of whether it was striatum, hippocampus, thalamus, or in the whole slide image, we did see a very distinct expression of genes. And we saw that the DRD3 and the CRNA2 seem to dominate in almost all regions. This was followed by GABRA2 and GRIA1, and then the pattern was similar in all the different regions of the brain. So this gave us a very strong indication about how the gene expression profile looked between different regions of the brain. And for the further analysis, we removed RBFOX3 because it showed very ubiquitous expression, being a neuronal marker. So next we performed the HiPlex neighborhood enrichment analysis. So we wanted to look at how these neighborhoods are enriched, how these neighborhoods of cells are enriched, and how they are related to each other in terms of the receptor gene family. So as I mentioned before, we had the different receptor gene families, and when we performed the neighborhood enrichment analysis and mapped it back to the whole slide image, what we saw was there was clear enrichment of these receptor genes in different regions. For example, the dopamine receptor could be predominantly seen enriched in the cortex and in the cerebellum. So we wanted to look at the enrichment neighborhood enrichment analysis. And first we did the UMAP clustering, and what we saw was that these clustered amongst themselves. So there was clear enrichment of these receptor gene families. And then we looked at the neighborhood enrichment analysis for positive and negative, for positive and inverse spatial relationships. And we use the Z-score method, which is a measure of over or under representation of a cluster pair. So here, as you can see in this neighborhood enrichment analysis, we would like to point out to a few neighbors or the neighborhood enrichment that came out to be very distinct. So if you see the AMPA receptor gene in the top panel, you'll see that there's more or less a homogeneous distribution, and they don't seem to be associated with any other receptor genes. Whereas the acetylcholine receptor was shown to be negatively or inversely enriched with dopamine receptors. And when you try to look at the colors here, and you can see that that's what matches. Similarly, with the GABAergic receptor neurons and the GABAergic transporter neuron, there seemed to be an inverse correlation. So this gave us a very good indication of the neighborhood enrichment, and we could predict some functional importance of these neighborhoods based on how they are, why they are enriched in the specific regions. So to summarize, using the highly sensitive HiPlex assay and the RNA-protein co-detection assay, we established a spatial gene expression map for visualizing GPCR family receptor within the normal mouse brain at single-cell resolution. We could identify cell-type specific gene expression of opioid receptors in different regions of the brain, and we could predict some functional significance of different cell types in the pain pathway. This study provides a deeper insight into understanding the spatial crosstalk as well as functional significance of different cell populations within various regions, thereby leading to a broader understanding of disease pathology. And in future, HiPlex V2 and ICW can be leveraged to investigate spatial expression profile of RNA and protein targets in pathological brain tissues for translational resource for pain and neuropsychiatric research. And I would like to just conclude by showing the audience that there are currently over 7,000 publications, and it shows the growth and adoption of the RNAscope technology over the last 10 years. Since we offer a wide range of assay to different biomarkers, we have been adopted really well, especially in the neuroscience field, which is our top application area. And we have currently over 7,000 publications in the top tier journals across different research areas. Thank you for your attention. You can visit us at acdbio.com, or you can contact our support at support.acd@biotechne.com.
Thank you, Dr. Basak, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. Now let's get started. Our first question here, is the HiPlex assay manual or automated?
Thank you for the question. Currently, the HiPlex assay is in manual format only, and we are currently working on a platform where you can semi-automate the HiPlex assay. So we'll be coming up with a semi-automated HiPlex platform, but for currently, we have it only in the manual format.
Great, thank you for that. Our next question here, how many targets can you visualize using the HiPlex assay?
Yes, so for FFP tissues, we can visualize 12 targets, up to 12 targets for FFP tissues. And if we are doing fresh frozen and fixed frozen tissues, we can do up to 48 targets. And that is using a HiPlex up reagent. And within the same section, you can go up to 48 targets.
Thank you. And a related question, can you perform image analysis with these assays, as well as which platforms can you do?
Yes, we can definitely perform image analysis with this with HiPlex V2 assay. We use Indica Labs Halo Itch module, as well as other platforms like QPath or ImageJ, which are open source. So you can perform image analysis and downstream analysis as well using any of these platforms.
Thank you. Another question here. How many antibodies and mRNA can you simultaneously detect in the ICW co-detection workflow?
Thank you for that question. Yes, so you can perform co-detection of one antibody along with 3 mRNA targets in a 3-plex or 4-plex RNAscope assay. So the maximum number of mRNA that you can do is 3 and 1 antibody, or you can do 1 antibody or 2 mRNA targets depending on your experiment.
Great, thank you. Thank you so much, Dr. Basak. Do you have any final comments for our audience?
Yes, I would like to thank LabRoots for inviting me to speak today. I have thoroughly enjoyed this session, and I look forward to connecting with all of you again in the future. If you have any questions related to the presentation or related to our other assays, please email support.acd@bio-techne.com, or you can visit our website for more information at acdbio.com. It's been a pleasure speaking here at this meeting. Thank you.
Thank you so much again, Dr. Basak, for your time today and your important research. Before we go, I'd like to also thank the audience for joining us today and for their questions. I'd also like to thank our sponsors, ACD Bio, a Biotechne company, for sponsoring today's webinar. Any questions we did not have time for today or those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until March 8th, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.